All right, let's go over a couple of parts of the cell and talk about what they do and what their function is. So the first one, I guess the most important one, is the nucleus. It has the DNA in it, and it's kind of like a safe where it holds the genetic material, and only a few things can go in and out. So can you see these little holes? They're designed for some proteins, and they're designed for some hormones to be able to come in, and a few things to come out, which is called RNA, which we'll talk about way more in the future. And then this is really just another protected layer for the RNA. The RNA is kind of fragile. UV light takes it out, some chemicals, viruses definitely want to get in there and take over. So we have to protect it, and if we didn't have that same protection, we definitely couldn't be around as long as we would like to be. Our next thing that we're going to talk about is ribosomes. I'm going to go back a picture just to show you two things. These little dots that are all out here, those are the ribosomes, and this folded material that's on the outside of the nucleus is called the rough ER or endoplasmic reticulum, and those are the next two things that we're going to talk about. In prokaryotes, the function is basically of a ribosome is basically to make protein. So it's like a little factory where a little bit of instructions come in from the DNA, which we're going to talk way more about this in the future, and basically turns that instructions into a product. And that product is going to be just proteins that we can do something with, or a hormone or something like that. These ribosomes, if they were free-floating, they'd be really hard to find. The product that this thing makes, called mRNA, only lasts for about 20 minutes. So if it didn't find a ribosome and wasn't able to be made into a product pretty quickly, what would happen is it would degrade and no product would be made. So that would be a major problem. So what we do is we house them on this thing called endoplasmic reticulum. And there's two different types. There's a rough type and a smooth type. But the rough type is called rough because it has all those ribosomes attached to it. It functions to make sure that we have a one-stop shop where we could come in, get the ribosomes, the message goes to the ribosomes, we can start making our protein, and then from there we can make it into a vesicle and send it to wherever we need to go. The rough ER, like I said, is made up of two parts. There's the studded part, or the rough part, and that's going to be for protein synthesis and packaging of that protein. And then there's an area that's smooth, and I'll show you a picture of it in a second. And it has no ribosomes. It's basically instead for lipid synthesis. Remember that lipid bilayer? It helps make that. It helps with some metabolism. It does a little bit of cell detoxification and makes vesicles. So let's see it. So here's the nucleus that we talked about. And right on the outside, this is what's blown up. Is this purple area is the rough ER. See, it's got all the little dots on it. Those are ribosomes. And the smooth ER is smooth, has no ribosomes, and that's the thing that we talked about, just does lipid synthesis and some other vital functions. Detoxification of cells. When things leave, so here's the ER, so ribosome. It's starting to make that first layer of protein synthesis. Remember that primary structure? So a little bit of instruction came in from the cell, and that instruction is helping make this primary structure of protein synthesis. So these would be the amino acids put together in the right order. Eventually what they're going to do is fold to make those alpha or beta pleated sheets, and then finally they're going to go ahead, and that would be the primary. The secondary is not shown. This would be the tertiary. And finally, if it needed extra pieces, it would get added. So protein 1 and protein 2 and protein 3 would come together, and that would be the quaternary phase. Eventually what happens is when this protein is done, we add these little sugars to it, and they basically are instructions. So they're either going to say, hey, I still need to add a couple of sugars to this, a couple of proteins to this one, or it could just be like, hey, this is where I need to go. So it could be a zip code to say this protein needs to get delivered to wherever, or it just needs some more parts. Eventually what happens in the smooth ER is we'll make this vesicle, and it will float to wherever it needs to go. So let's just kind of get a big picture. Nucleus, genetic information. Some genetic information comes out of that to the ribosome. The ribosome starts making those protein levels. The protein levels then eventually are going to get directed in a little vesicle, so like a delivery mechanism, to our next area, which is going to be called the Golgi apparatus. And the Golgi apparatus is like the UPS of the cell. It's going to deliver. It modifies and packages proteins. So let's say protein A and protein B and protein C have to come together. That's where we're going to put it together and package it the way it needs to go. And then finally it's going to deliver it. And those proteins are coming from the ER, but specifically it's going to come from the smooth ER. Remember, it starts being manufactured by the rough, delivered by the smooth, and it's delivering it to the Golgi apparatus. And then it sends it out wherever it needs to go. It can either go in the cell or it can go outside the cell. So if we get the big picture, nucleus, genetic information, ribosomes, they're going to start making the proteins. Once the proteins are sort of done, at least that protein is done, it's going to go to the smooth ER, get delivered to the smooth ER, to the Golgi apparatus. So smooth ER to the Golgi apparatus. The Golgi apparatus will package, so let's say it needed protein A, protein B, and protein C and put them together. And then finally what it's going to do is it's going to deliver it. And it's going to deliver it in this little vacuole. And it basically is either going to secrete it outside the cell, or it's going to take these vacuoles and move it somewhere else in the cell. Whatever that protein needs to do and wherever it needs to be delivered, this UPS system is going to do that. We have some other vacuoles that our body has. We're just going to focus on the first one and the last one. This next one is just kind of for your own information. Lysosomes are found in animals, and peroxisomes are found in us as well, and both of these serve to do different things. Lysosomes basically are our recyclers. We're done with a protein, we're done with something, and we want to get some amino acids or whatever out of it. We're going to put it in the lysosomes, and we're going to recycle it there. Peroxisomes are a little different. What they're going to do is anything that's foreign or suspect, that it might be, let's say, a virus or something that we just don't know, we're going to put it in the peroxisome and kill it. So when I think about it, these two are like the garbage cans of the cell. We have a recycling bin at home, and we have a garbage, the one that just goes to the trash pile. That's what these two are. We're going to be able to recycle right in the cell, or we're going to just kill it, and eventually just spit it out into the bloodstream. So basically what happens is this could be something from outside the cell or inside the cell. It goes into the lysosome. The lysosome is going to digest it, try to use as much as it can, and then basically eventually just spit it out into the cell for it to use. The lysosome could also be used to kill things. So let's say this mitochondria died. So the lysosome will come, sort of bond with that mitochondria, sort of dissolve it into anything that we can use, and then spit that out to the cell to do whatever it needs to. Remember I told you there was one more vacuole? I said you're definitely not responsible for the exam, and I definitely am saying that. It's called the central vacuole, and basically what it is, it's in plants, and it stores water. So when a plant runs out of water, the central vacuole sort of collapses, and when it collapses, that's why we see plants sort of leaning over. And then when you feed a plant, all the central vacuoles fill back up, and that's why the plant goes back to being sort of perky again. And then, of course, our peroxisomes, we've already talked about them. They're similar to lysosomes, except these are going to be killers. They also help break down some fats if we ever need them to. Detoxifies alcohol. Alcohol is really bad for us, so this is where they'll go to sort of break that down. So here's the big picture. We have the nucleus. We have our ribosomes. That's going to be on our rough ER. Eventually we're going to get to our smooth ER. That's where we're going to deliver it to the Golgi apparatus. The Golgi apparatus is going to go ahead and package it, and so add other proteins that need to be there, and then finally deliver it either outside the cell or somewhere inside the cell. And of course we have other vacuoles like lysosomes and peroxisomes that are there to do either recycling or killing of foreign bodies. In our next video, we'll talk about the mitochondria.